Hi, everybody, and welcome to the Sierra Club Airport Marina Group's first online long-distance Zoom meeting. I'm Richard Harmel. I'm a member of the Airport Marina Group Executive Committee, and I'll be your host this evening. Restoration ecologist Dr. Margot Griswold has agreed to share her insights on what restoration really means.
Welcome, everyone, and thank you for tuning in. And so what I wanted to kind of build on the last speaker who talked about soil and how wetland restorations are not living up to what they should or could, mainly because lacking of soil components. And so I'm going to touch on that as well as I go along because soil is everything. My company's name was Earthworks, and our motto was from the soil up. Without the soil, we couldn't do anything. So I learned a lot about the soil in the early years, and so I feel lucky. Even though I'm not a soil scientist, I've worked with a lot of soil scientists. So everything I talk about tonight, you have to understand, is always a team effort. There's always many different people working on these projects with different expertise, and I'll remind you of that as we go. And so I want to reflect on what habitat restoration is, and an approach we can take to restoration and enhancement of Ballona wetlands. And the reason I want to focus on this is because I think people throw the term around, and I'm kind of precise about how I like it defined. And not only that, but the state has a precise definition on what you can do in wetlands, and restoration is all you can do in wetlands that are within the coastal zone. So I think it's important to understand what restoration is. So it is the process of intentionally altering a site to establish a defined indigenous historic ecosystem. The goal of restoration is to emulate the structure, function, diversity, and dynamics of the specified ecosystem. So it has to have been there recently. Creation, on the other hand, establishes a historic ecosystem, so it could be somewhere in California, in Southern California, but it never was on the land, so it was not supported where one person is trying to do this particular habitat. So creation establishes a historic ecosystem on lands that did not previously support that ecosystem. I didn't make this up. It's from the California Society for Ecological Restoration. But it's what I lived by. And just for the record, I've done restoration, I've done revegetation, I've done land reclamation, and I have done some creation. And I can tell you that I would never do creation again. It's very difficult and takes a very long time and it's just not satisfying. Very difficult. So I mentioned revegetation. What's the difference between revegetation and restoration? So revegetation establishes a historic native habitat on a drastically disturbed site with no natural soil horizons, so no organic A, B, C horizons in the soil, no soil ecosystem, and little to no seed bank. Whereas restoration establishes a native habitat that once, more than likely, occurred on a site with more or less natural soil horizons, a disturbed soil ecosystem, and an infinite weed seed bank, generally. So it's a degree of disturbance when you're using the term revegetation versus restoration, and I'll show you some examples. So this is an example of revegetation. So here we have a roadway being built through an open space in Orange County, the 73 toll road, which I was the project biologist to manage the mitigation and to make sure that all the regulations were followed. And so this is the soil on a slope in 1994. You see it is nothing but subsoil brought to the surface, filling Bomber Canyon by a lot. 1995, the slopes are built. This is 4 benches. Each slope is 70 feet long, or high, I should say. And then a bench, and then 70 feet. So that goes down 4 big benches. And I'm standing at the bottom taking this photo. We've now seeded the site, and I used some of the brush that was cut prior to building. We set it aside, and we put that at the toes of each of the benches. And so here you see in 1997 what the seed mix has done. So this is what I term the living mulch. This is a native grass that we seeded heavily, along with all the other seeds we were putting on. So we put our pioneer species, our grasses, our forbs, subshrubs such as deerweed, and also what our target dominant revegetation would be. All seeded at the same time, but what comes up is, in the first few years, just the pioneering species. And that's following a succession model based on observations from fire. So it doesn't happen instantaneously. 1995, we seeded. 97, you see what it looks like. And by 2001, this is the habitat. So after 6 years, the target shrubs began to dominate the slopes. So what's restoration then, if that's revegetation? Well, we can define restoration as a reestablishment to return historic natural functions to a site as previously existed, or rehabilitation to improve the general suite of functions of a degraded site. And usually rehabilitation is less disturbing than just reestablishing. So let's look at West Bluffs for restoration. So these are the slopes, you know, above to the south of the Ballona Wetlands. They're a sandy bluff. And this is what they looked like when we started the restoration. It was mainly wild radish, short pod mustard, and annual European grasses, with a shrub here and there to give us an idea what it might have been. Research into the historic notes and photos of what had occurred there previously gave us the indication of the coastal sage scrub slopes. So again, following the succession model, we planted all the species at one time. We seeded them after preparing the slopes. So in that revegetation project, you saw how bare the slopes were because the soil had no seeds in it, and we had a blank slate to start with. We added everything, like an artist. Here, there's an infinite amount of seeds in the seed bank. So we had to do site prep for a year or two before we would seed. And once seeding occurred, then the usual pioneer species came up as we expected, but we got an unexpected boost. If you see this gray-green plant all over the slope, that is Nathalium, pseudonathalium, I think they changed the name to, microcephala. It was not in our seed mix. We had pseudonathalium californicum. But this was on the slopes. We didn't see it before we started. It was being suppressed by the wild radish and the mustard and grasses. But as soon as we controlled those, it jumped forward. So, and then this is the development, as you see in the second and third year, we start getting more of the shrubs. A few dry years, and 2010, I think, was a good year. At any rate, we achieved the performance here, and this is in 2020. So it's still looking even better than it did in 2010. So that is simply weed control, no equipment except for weed whackers to take out the non-natives before they flowered, raking off the thatch so we had a seed bed, and then seeding. And then, you know, several years of hand weeding followed. That's an ecologically efficient way to restore. No spray, no weed whacking once you put down your seed. It's hand weeding. It's hard work, but it pays off. And this is just a video of the whole slope from Cabora Drive. We did have some revegetation on the West Bluffs as well. This was filling Hastings Canyon outside of the coastal zone. Within the coastal zone, there was no filling allowed. No bulldozers could go on the slope in the coastal zone. I'm going to repeat that. The Coastal Commission wouldn't allow any bulldozers to go on the slope of the coastal sage scrub, degraded coastal sage scrub slopes. But outside the coastal zone, they filled the Hastings Canyon erosion feature, and we then had the clean slate of a revegetation slope, and this is the development, much as you saw in the earlier photo. And I would like to point out that we had, here's the manufactured slope, the revegetation slope in the background. This is the restored area here. And at the last minute, the Coastal Commission threw another 12 lots at us. And that's here. And this is an area that was added and didn't receive the same pre-treatment in the same years, in the same intensity. So you see that without that site preparation, it's difficult to achieve the type of restoration you really want. So here's a general trajectory of habitat development that you see whether you're in revegetation or restoration. So here we have year, and here we have percent cover on the y-axis. And you see that pioneer species start out, they jump to the fore, they germinate almost instantly, and you have a good cover for erosion control and to protect the soil from raindrop erosion as well. And meanwhile, your target species, in this case, we've been looking at coastal sage scrub. those target species are developing so that by around year 6, you start really seeing that, and the pioneer species are falling out because of shading. However, they've all seeded. So if you had a catastrophe such as a fire, you would have your seed there, your pioneer seed would kick in after the fire, just like you see in nature after a fire. And we've seen this happen in some of our projects that have burned after maybe 10 or 15 years. They come back without any seed being added to develop after a fire into the target habitat. So this is our generalized model. Let's see. There are other components that come later after the 6 years, from 6 to year 15 or so. And here's a Dudleya multicollis, which is a rare species in Orange County that was added and came up later. This is the Nathallium, and here's Nathallium, Pseudonathallium microcephala, who was not on the West Bluffs slope, but came up after being released from the mustard domination and the wild radish. Also, Pseudonathallium will take years to come up in a re-vegetation area. So because there's no soil development and it needs a particular soil companion to develop, it does not appear until after around year 10. And so that brings me to the secret ingredient of all revegetation, restoration, you name it, in the soil are the mycorrhizal fungi. Now they're tiny, as you might think from mycorrhizal. There's 2 types. There are ectomycorrhizal fungi and arbuscular mycorrhizal fungi. So the ectos are a lot on trees, oak trees, pine trees. Many trees have ectomycorrhizal fungi, and as you might think, they're on the outside of a plant root. I'm showing in this diagram the arbuscular mycorrhizal fungi, which actually goes into the root of a plant between the cells, and what the mycorrhizae do, but whether they're ectomycorrhizae or arbuscular mycorrhizae, they act as a extension of the root system for plants, and they absorb water and they absorb nutrients, especially phosphorus. Now, this is really important in Southern California because our soils are very low in phosphorus. And that's, as you know, one of the 3 main nutrients for plants. So mycorrhizal associations help 80% of plant species. 80% of plant species. That's not just on the dry uplands, but also in wetlands. Recent studies show that all wetland types support mycorrhizal fungi. So any kind of wetland, a freshwater marsh, a saltwater marsh, you name it, there will be plants there that form a mutualism with these microfungi. So I say a mutualism. I've told you what the fungi give the plant. They absorb water and phosphorus, but the plant gives the fungi its food, because fungi are not autotrophs like plants. They can't make their own food. So it is a mutualism, costs a little something for the plant, and that's how mutualisms work. You get something, you give something. also, I think it's very interesting that recently they've found that the Ectomycorrhizae, and I believe the Arbuscular as well, are thought to be like the soil internet between plants. So this is how plants can communicate. They set up a net. So these hyphae go not only into the root, but they go between the roots. And especially in the arbuscular mycorrhizal fungi, they aid in maintaining soil structure. So they have a protein called glomalin, and that forms a glue-like protein. And along with humic acid is really thought to aid in soil structure, the formation of soil, and the structure of soil is very important to oxygen. Oxygen is needed for the roots of all plants. So you have to have pores in the soil for oxygen to be there. Without that, you get compacted soil. And we all know nothing grows in a dirt roadbed that's driven over and over, mainly because it's compacted and there's no oxygen for plant roots. So AM fungi are extremely important. Help reduce the soil erosion because of that glue-like glomalin. And certain of the early successional plants in our model that we use have a very strong association with AM fungi and vice versa. So you want to favor seeding many of those early successional to build your microfungi net, and then you have a great ability to have a very good soil, absorbs water, holds water, and aids in plant development. So another really interesting thing, and I witnessed this, I observed this myself. There would be sites where we had weeded and now our plants were very well established, and I would see this army of mustard standing exactly at our boundary, as if there was a wall. And I thought, well, that's funny. Look at that. Why is that? And I started reading and asking questions, and some of you may know Ted St. John, who was an early proponent of using mycorrhizal fungi in restoration, and I learned a lot from him. He said, well, I think they're antagonists. And sure enough, I was looking around for things to put on the presentation tonight, and here was a non-mycorrhizal plant. Well, all mustard family is not mycorrhizal, none of them. So don't add mycorrhizae if you want to grow radishes in your garden. It won't help them. So apparently, here we have the net in the light gray. That's aiding in the roots of these other plants, while meanwhile, there's antagonism going on and inability to absorb and an inhibition of root physiology. So there you have it. Mycorrhizae helps us fight the weeds in particular types of weeds. Unfortunately, not all weeds are not mycorrhizae. Is that the right way to say it? Anyway, if anyone has any questions here, I'll stop at this slide and you can ask.
What is the timeline for the buildup of the microfungal?
So the timeline is, in a restoration, usually you have mycorrhizae in the soil because you haven't drastically disturbed the site. And so I rarely have to add mycorrhizae when doing a real restoration. You do add some, especially if you're putting container plants in your site, because they'll come inoculated. Mm. It's very rapid. We did tests on the slope I showed you on the toll road. I had Irvine Valley College students out, and we took root samples and we measured the distance from the container plants, because in those days we were it was dogma that you could not hydroseed mycorrhizae with your seed. You had to add it in an inoculated plant. And so on that first year on that project, we planted liner plants 10 feet on center of stipa to inoculate the soil. And then we measured that after a year and a half. And we found that the mycorrhizae had moved approximately 1 meter. And it moved from that inoculated plant because we had seeded with very friendly mycorrhizal pioneer species that had come up immediately in the first year and the second year. Mm. And so the roots of all those plants were moving the mycorrhizae through the soil. Mm. So it's quite fast. If you take topsoil, which we did, we were able to get some topsoil on this project. On another part I'll show you momentarily, you can get immediate results in the topsoil. Every plant you sample that is mycorrhizal will have mycorrhizae in the first year. Mm. So that's the best way, but to take topsoil means you have to take it from somewhere. We were working on a road project, so it was a balanced cut and fill, so we could take where they were taking out good habitat and store that topsoil, crush the vegetation and store the topsoil.
Thank you for that. We have a couple of questions that are referring to how do you protect wildlife, and how does the recovery of wildlife species intersect with restoration of a plant community? That's from Marcia. And then another question about how do you protect the insects, birds, and animals when you do a restoration of ecosystems, and what happens to these species if it takes up to 6 years to revegetate?
Well, they didn't have any habitat there.
Yeah.
So many species you're restoring, so the species nearby are going to have that coastal sage scrub habitat. So what you have is wild radish, annual grasses, and you are going to take that habitat for that 1-2 years of site preparation. So you won't have the same amount of habitat for your skippers or bees that might be using that radish. Because it's a restoration and we're not doing anything in the soil, all the soil organisms are fine. And in fact, you saw the seeds of the pseudonathalium is released, actually. So you're benefiting that species. We didn't have any loss of gophers. I can attest to that. In a restoration, it's more minimal disturbance. In a revegetation, because it's a project that drastically disturbed the site, it's the project that disturbed the soil, and so you're putting it back. When a project like that goes through, it is devastating. It is one of the hardest things I've ever had to manage, trying to move snakes, lizards, squirrels. It's the only project I did that I had to do that. But so in the restoration, it's easier. There is a time frame of which that first season you're going to be getting, all the plants are going to be flowering, so you're bringing back bees, and you might have some more native bees rather than the European bees. So it's a fairly short time in a restoration if you're doing a successional model. However, that depends on how long you have to treat the weeds. And so when you're out there cutting the weeds and then the rains come and then you cut the weeds again and then the rains come, that could go on for 2 years or so. So depending on the size of your project, you would phase that work so that there was habitat for even the species that were using the less desirable target, less than the desirable target species.
Okay, thank you, Margot. I have a couple more questions here, and then we need to move on. Marcia wants to know, first of all, not just protecting, but the recovery of wildlife. And she's also wondering, shouldn't we or whoever is planning restoration be asking questions like, what does the least Bell's vireo, the endangered songbird, need for recovery?
Well, yes, and I'll get to that in the next slide. These projects I was showing you were for coastal sage scrub species. So least bells are going to be in the lowlands rather than on the slopes. Blue-gray gnatcatchers, I was hearing those within 3 years on the West Bluffs. On the toll road, we've had within the 4th year, we could start hearing California gnatcatcher, which was the target species for that habitat. So you can get a fairly quick recolonization. And in the next slides, I'll talk a little bit about that. I hadn't planned to, but I can.
Okay, moving on. Thank you.
Moving on. So restoration can be divided into 2 activities, reestablishment and rehabilitation. And the reestablishment returns the historic or natural functions to a site as previously existed. And rehabilitation improves the general suite of functions of a degraded site, usually resulting in less disturbance. So I would say the West Bluffs were a rehabilitation in terms of restoration because there was less disturbance. So just a quick look at Ballona this year, and I guess we know it holds water. Here's the Area B up near where they used to drain the water out. Those drains have been plugged. Here's the salt pan. And here are some small pools along the way to the salt pan below the current freshwater marsh. So this is what was predicted by the historical ecology of the Ballona Creek watershed, 2011. So the more recent science indicates what Ballona should be if you're going to restore Ballona, if you're going to enhance and rehabilitate and reestablish. It never was a full tidal wetland. And I hear people talk about historical ecology like, oh yeah, well that's just one T-sheet, or you can't base anything off that. Well, yes, it's not just one T-sheet. It's the compilation of the historical record using older maps, T-sheets, irrigation maps. All of these things go into account and assemble the report. It's not just one thing. It's many things, and it's a science. It is a science, and it's so valuable and should be used. You know, I've thought I knew what things should be, and I've been proven wrong, and it's, you know, everyone thinks they might know something, and then when they're shown the facts, you should be able to say, oh, I was wrong. Let's do it the right way. And I think that's what's kind of missing here in the approach to the Ballona wetlands. So again, more recent science, a classification, a model that shows how our Southern California estuaries are mainly closed and were not open. And I think looking at regionally, you can see in the historical, the acres of salt marsh, salt flat, which is our salt pan, seasonally flooded with fresh water. You can see what they were, and you can see the contemporary. So the change has been only 12% loss of salt marsh, whereas the salt flat or salt seasonally flooded is 90% lost. Brackish and freshwater marsh, 54% has been lost. Whereas, look at open water mudflat, that's the title. 615% increase. Now, this is a study from northern San Diego County, but you can extrapolate that if you just look at Orange County and what's happened there as well. And it's a homogenized group of estuaries now, fully open, and trying to keep them open is difficult because, as predicted in the model in the previous slide, they will close, and you have to spend your money keeping them open. So I think rather than turn Ballona into just another full tidal marsh at great expense, why not look at her and say, you know, what can we do more efficiently? What can we do that's more in line with the rainfall, the runoff patterns, the groundwater that needs to be protected? What can we do rather than come in here and dig it up? And let's start with Area A because they seem to have changed a bit their tune in areas B, south and north. So these are some of the species we see. And you can see that the center of Area A was most likely a wetland and not a tidal wetland. You can see we still have Fiji ditch here, which is a riparian, you know, it gets a tidal influence, but it also must be getting some freshwater flows just based on the vegetation in there. So the weed load, the non-native percentages, but bear with me because I looked at their more recent, they didn't actually present the data this way. And so I am using it this way. I went out and walked along the edges because I can't go in. No one can go in. So just walking along and looking at the vegetation. And I was looking for all the black mustard they said was in here. And I do see a lot of black mustard along Lincoln. And a little over here, but not too much. But mostly I saw short pod mustard here. And I saw a lot of chrysanthemum, some castor bean, and euphorbia. And I'll just say that we know how to beat the mustard. Addition of mycorrhizal fungi and mycorrhizal plants, seeds. Do some weed control of the short pod mustard. Chrysanthemum, I believe, based on my experience in the Baldwin Hills, has a very short-lived seed. So we don't have to fear the seed bank as we do with the mustard. That's good because chrysanthemum's a mycorrhizal plant. But we can pull that plant up easily, and it holds on to its seeds for a very long time, so it's very forgiving to get rid of. I think we've done a really good job just with volunteers in the Baldwin Hills in the last 10 years. Euphorbia, similarly, we've gone after that in the Baldwin Hills just by hand. That one you have to kind of cut off. Euphorbia and castor bean are related. Gophers don't like them, but they're kind of obnoxious. But you can get rid of them. And you can do it without chemicals. The other cool thing is there are these big areas of native plants, California sagebush, you have laurel sumac, and then of course the Lewis's primrose, which is an endangered species. Those are found there. So of course we have to do surveys before and map out where you will and will not weed, and if you're going to go near the primrose, carefully. We're doing a project right now in Laguna Beach where the crownbeard is, and so far your Sierra Club compadres there think we're doing a great job protecting those plants and getting rid of the mustards out of the area. So it can be done in an ecologically efficient way. And this is an example from Sandy DeSimone with National Audubon at Star Ranch, where she manages 4,000 acres. She uses no herbicides. This is artichoke thistle dried and already gone to seed. This is what her test plot looked like when she started. This is after a few years of cutting with weed whackers. You see the artichoke thistle here in its rosette form. It bolts, they cut, and they do this repeatedly for several years. And here they've seeded then finally when they have few enough artichoke thistles that once were a monoculture, and that's how her habitat developed. No herbicide. Just hard work though. But you don't get anything without hard work. So the design alternative that I would suggest is to base it on the most recent science from historical ecology and models of closure patterns and from citizen science observations, of which we have many. Avoid impacts to landscape features that have been stable for the last, at least 150 years or more, such as the dunes, the salt pan, and the brackish marsh habitats. Don't remove the levees. Keep those Ballona Creek where it is now. There's just some things you can't really change. What good does it do to release that water into this wetland in just that last mile? It's a strange idea, if you ask me. The creek never flowed like they're showing it in any case in the plan. The creek stopped up about Lincoln and it just sort of were fingers going out, much like you see Sentinella Creek now still flowing on the south side. I would reduce the tidal channels to avoid draining the brackish and fresh wetlands, because that's what tidal channels will do. And put in new tide gates if they need to actively manage to, create that diverse condition. And conserve and enhance the native biodiversity. So including the brackish marsh and meadow and tidal marsh, upland and rare endangered species. And these are all Jonathan Coffin's photographs by area. And it's just a lot of species, bird-centric, because I am Los Angeles Audubon. But there's also an amazing assortment of insects, or you wouldn't have the birds. There must be voles. There must be for the raptors to be feeding on, as well as other small birds. There are butterflies, skippers, and mentioned the primrose and the burrowing owl. So even up in C, the much maligned C, there's opportunities to create and enhance what was there, not create, but enhance what actually is there. Some wetland features that occur. We still have Fiji ditch coming through. So there's, I think, things that just haven't been explored to take advantage of, to do really an efficient, ecologically speaking, efficient restoration. And that's it.
Okay, that was great, Margot. You took us on quite a journey there. I have a few questions for you, and then we can address those and then continue on. Roy wants to know, why are only plants considered in restoration, and thus plant ecologists driving restoration? He says, why not recover early successional animals in early stages as well, such as insects, small lizards that use dead wood, rocks, perhaps small mammals, alongside the plants? I do not see animals being recovered later with restoration called complete without translocating animals.
Well, I think that depends on whether there are, in a lot of my sites, they have been natural areas adjacent. So in fact, they do colonize. And I know that because even on some of those revegetated slopes, that I wouldn't imagine, when we had to go back and put wildlife fencing, move it from the outer edge of the right-of-way right to the road edge for better protection, we had to be digging holes and to put in these great fence posts. And I was amazed after it was a 10 or 15 years what we would find, because we'd have to go every morning and make sure nothing got in those holes before they started doing the fences. And there were some interesting snakes, lizards for sure, in those areas. So I think it depends on what's nearby. And I agree, yeah, plant people are usually at the forefront, but I don't know, I work with a team of soil people, and we usually have the wildlife people telling us what the goals are, be that U.S. Fish and Wildlife Service or California Department of Fish and Wildlife. And sometimes it's lizards. Sometimes it's, what are those, spadefoot toads. Sometimes it's birds. Sometimes it's making connections for coyotes to use a habitat to get from one to another, such as using Bonita Creek to get to the upper Newport Bay, where they were needed to control red fox, who was preying on the clapper rail, or the formerly named clapper rail. So I agree, in some projects that is true, but in a lot it's a moving group of, why do many of these projects get done? They're done for mitigation. And so the target species come down from that, usually. But Ballona's different because it's not a mitigation. It is kind of pure that way if we kept it that way.
Huh.
So you could, these are all good points to bring up in the planning, because it's hard to preserve soil organisms when you're digging it out with a bulldozer.
Yeah. Leslie wants to know, why is the open water mud flat so prevalent / preferred?
You mean when people do these mitigations?
Yeah, I guess so.
That's why. Because, why are they so preferred? Because that was the target they had to replace. They were taking it out and they had to replace that somewhere.
Mm-hmm.
Also, I think it has like this. It has to do with people's perceptions. When you work with parks, they want trees. Shrubs aren't good enough. It doesn't matter. You explain many times that trees wouldn't live here. It's an idea people have that trees are better than shrubs. I think the same. If you talk about a coastal wetland, what do most people think? A tidal wetland. They don't think of a coastal wetland being a freshwater wetland, a freshwater marsh, a brackish marsh. They would think the salt pan was there because of the tides, instead of the salt coming from water evaporating from this slightly raised area, evaporating and leaving the salts behind. And they would be amazed at the life that's in that. To Roy's point, the life that comes out of the salt pan is incredible. So when I look at a plan that sprinkles salt pans around like they were, willy-nilly, they don't understand what's in the salt pan. They would have to take that soil, they would have to inoculate everything, destroy it it doesn't make any sense. But anyway, that I think is why they tend towards making more open water, full tidal wetlands, because it's somebody's idea that that's better, more productive. But I think what is not yet well understood is the role of microorganisms, soil organisms, microfungi in sequestering carbon. They're extra roots. I think you would see people thinking differently about it if they thought these other wetlands were as productive or more productive in terms of sequestering carbon. So it's a bit of a two-way thing. I think mitigation drove a lot of stuff, open water for ports, port projects,